Hello students, we will study today a family of viruses whose phylogenetic analysis based on to the DNA fingerprinting showed similarity with the chimpanzee's virus. It is now believed that the humans must have acquired it from chimpanzees about 1.6 million years ago, and that's about the rise of the modern human life about 200 years ago. Uh, the term was first applied in the literature back in 17th century, but we did not found the name of this virus until 1945. It was 1945 when the first name of this virus, they belong to the family HerpesVirus, came into the literature. Now, the HerpesVirus family comprises of herpes viruses, and it is a group of 6 viruses, starting from herpes simplex 1, herpes simplex 2, varicella zoster, cytomegalovirus, Epstein-Barr virus, and herpes simplex virus 8. So we have the HSV1, HSV2, VZV, CMV, EMB, and EMB, and HSV8. 6 important members of the herpesvirus families comprising of herpes simplex 1, 2, 8, along with that of the varicella zoster, cytomegalovirus, and the Epstein-Barr virus. All these viruses, they are structurally same, and they have icosahedral symmetry. If you look at this, and all of these viruses, they cause blisters on the skin, and they can cause some of the nerve damage, and they can also cause some other diseases that we will discuss. So let's first see the architecture of the generalized structure of the herpes virus. Now, as we know that the virus is icosahedral in symmetry, and we all know that we have almost, you see the packets of 20s, different angles of at least 12 facets, and that make this the icosahedral symmetry. We have the double stranded DNA viruses, and this is what we call as the nucleocapsid, which make up the capsomers, which is protein in nature, and along with that we have the lipid bilayer. The virus is an invert virus, that we have a lipid bilayer. Now, in between the nucleocapsid and the lipid bilayer, we have a material that we call as the tegument. This is a specialized structure which is found in this virus. Now, along with that, we have these different spikes. We have the surface glycoproteins, what we call as the GD, we have the GB, and we have the GDL, and we have the surface glycoprotein GC. So there are various components of the glycoprotein which are present in the form of spikes on the surface of the, on the envelope of the virus. The virus is a double-stranded icosahedral virus, and it has a double or bilayer envelope, along with the in between the nucleocapsid and the viral bilayer is a material what we call as the tegument. So all structures, all of these viruses, all these 6 viruses, they are structurally same. They are all icosahedral, but they have affinity for the different specific cells in the body that we will discuss in the coming slides. So 6 viruses belong to the family herpesvirus, are clinically and important from the examination point of view. The herpes zoster, the herpes simplex 1, 2, 8, along with that of varicella zoster virus, cytomegalovirus, and Epstein-Barr virus. Now, the replication cycle of all of these viruses, as it has DNA viruses, so as we know that all DNA viruses, they replicate in the nucleus, and they form internucleated. The virus has shown the greatest tendency towards the latent infection. With the initial infection, with the acute infection, the virus later on become, you see, quiescent in its action, and it lies vacant in somewhere in the tissues. But the reactivation of the virus can occur because of some of the immunosuppression or other factors that reduce the immune of the human body, and the replication would start. The virus can broadly be divided into three sub categories based on to the effect, what we call as, which has the alpha subgroup, we have the beta subgroup, and we have the gamma subgroup. So we have three subgroups of the herpes variety families. The alpha group that comprises of the herpes simplex 1, herpes simplex 2, and the varicose zoster viruses. And this group of viruses, it has the killing effect on these cell lines, or the human tissue cell, and they causes vesicular formation or vesicular rashes. The second group is, which is less pathogenic, and it does not cause the vesicle formation or the rash formation on the human tissues, and it comprises of the cytomegalovirus virus that belongs to the beta group, and the Epstein-Barr virus that belongs to the gamma group. So three subgroups of the herpes viruses. Alpha group comprising of herpes simplex 1, 2, and varicose zoster viruses, they causes vesicular rashes, whereas the less pathogenic virus like cytomegalovirus belonging to the group B, Epstein-Barr virus belong to gamma groups, they do not cause the vesicular rashes. Now, all the three, HS1, 2, VZV, and cytomegalovirus, they form multinucleated giant cells. This is a characteristic of all these viruses. What happens? They cause the abnormal cell division in the epithelial cells, and this creates, you see, cytomegalovirus, which is much larger than the normal epithelial cells, and it contains multiple nuclei. This is what we call as the multinucleated giant cells. They have the multiple nucleus, and they are larger than that of the normal epithelial cells. Now, this is how the multinucleated giant cells will look like if you make a smear from the lesions and you stain it with the help of different staining procedures. And we mostly use the methylene blue stain or the ginseng stain or the right stains. And because this technique was developed by a scientist whose name was Zhang, so it is called the Zhang smears or Zhang test. Now, this is the microscopic appearance of the multinucleated giant cells, which is illustrated by the Zhang smear. You take this smear, you make the smear from the lesions on the skin, and you stain it with the help of either iodine stain, or you can go for the methylene blue or white stains. Now, the smear or the scraping of the ulcer base, how it look like, you see, this is 1, 2, 3, 4, 5, 6, 7, 8 nuclei, the multinucleated giant cells that we can see with the help of Zhang smear. So if you have a history of somewhat vesicular lesions on the skin, and if you take this specimen or make a smear from the skin lesions and you stain it with the help of what you call as the ginseng stain or methylene blue stain or the rice stains, and this technique is known as the Zhang's techniques, you can see the multinucleated giant cells in these smears. So viral infections having vesicles, smears with the help of Zhang stains, so the multinucleated giant cells, they are certainly can give you a best clue towards the infection with the herpes simplex virus 1 or 2. Now, the general properties of the herpes simplex virus, broadly speaking, we can mainly distinguish by the antigenicity and the location of the lesions. Both of these HSV1 and 2, they are antigenically different. This means that the immunity against one cannot provide you protection against the 2, or the vice versa. And based on to the location of the lesions, the HSV1, herpes simplex 1, they form lesions above the waist, but mainly in the oropharynx, the face area and the oral cavity. The herpes simplex 2, they form lesions below the waist, particularly in the male and female genitalia organs. So broadly speaking, HSV1 infect the above the waist, and the HSV2 infect below the waist. So in the below the waist, we have mostly lesions in the genitalia, and in case of one above the waist, it's mostly the face and the oral cavity that is being involved in herpes 1 infection. The herpes simplex viruses, HSV1, it causes almost 8 different diseases, starting from acute gingivitis, recurrent herpes labialis, what we call as the cold sores, keratoconjunctivitis or the keratitis, encephalitis, And all of these diseases, they occur mainly in adults. In adults, all of these, the 1, 2, 3, 4, 5, 6, and we have 1, 2 more. So many of the different involvement of the different organ of the body, they give you different pictures. So HSV1 causes almost 8 clinical diseases compared to that of the HSV2 that mostly involve the herpes genitalis, that is genital herpes, and it is mostly also caused the neonatal meningitis or encephalitis and other non-neonatal herpes diseases and aseptic meningitis. So in case of, you see, herpes 2, it is mostly involved the genital area, or it can cause the nervous signs in the children. Now, infection by the HSV2, it is a common cause of what we call as the erythema multiforme. This is skin lesions caused by the infection with the 1 and 2, and we will see that how they look like. So, let's compare the both of these diseases before we start with the detailed discussion onto the HSV1 and HSV2. So broadly speaking, we can compare both of these diseases with reference to the etiological agent, with reference to the transmission. Either they go into latency or not, and what kind of lesions they will produce on the skin, what are the complications, and what are the dissemination in compromised, immunocompromised individuals. Now, the etiological agent, as we know in case of HSV1, is for the herpes labialis, pharyngitis, gingivostomatitis, keratoconjunctivitis, encephalitis. They all are caused by the herpes simplex virus 1, whereas the herpes simplex virus 2, HSV2, it causes herpes genitalis, both in male and in female. The transmission case of HSV1 is through close contact, usually by contact with the face, whereas in case of HSV2, it is sexual or the close contact information that causes the transmission. The latency in case of herpes simplex 1 disease in the trigeminal ganglia, the sensitive trigeminal ganglia, and it makes sense because the herpes simplex virus 1, it causes infection in above the waist, mostly in the face and the oral cavity. So therefore, the virus, it goes latency, remain quiescent in the trigeminal ganglia, and whenever there is immune system reduction, our body immunity is compromised because of the diseases, because of the medication, because of the viral infections, the reactivation can take place. It may take even years and years to reactivate the virus. So in case of HSV1, the latency of the virus is in the trigeminal ganglia compared to that of the HSV2, which causes lesions below the waist, and it is primarily the sacral ganglia where the virus undergoing to the latency. The skin lesions in case of HSV1, they are vascular lesions, and they of course will appear above the waist, in the face, in the mouth, compared to that of the vesical lesions caused by HSV1 that will appear below the waist in the internal or external genitalia, in the bucket, and on the thighs. Now, the complications of HSV1, it is a bit low, what we call as among the personnel working on the oral cavity. This is mostly with the physicians or with the dentists who, without wearing or going under precautionary measures, they manipulate in the oral cavity and they incidentally or accidentally come in contact with the lesions and they got the infections. In case of HSV2, because it is deals with the genital organs, or this is mostly among the obstetrics and gynecologists who deal with all these cases. The neonatal encephalitis, which is another complication of the HSV1 and HSV2, about 30% of the cases of the HSV1 infection, they have the neonatal encephalitis. Whereas most of the children who have infection with the HSV2, they will have the encephalitis. So in both cases, the weight loss and neonatal encephalitis, both of these complications, they are seen with the HSV1 and HSV2, concerned with those individuals who are working with the oral cavity or with the gynecological and obstetrical procedures. And encephalitis in children is more common with HSV2 infections compared to that of the HSV1 infection. The disseminated infection in immunocompromised individual can occur with HSV1, but it has been rarely reported with HSV2. So forget about the disseminated infection because it is always common with that of the HSV1, rarely with the HSV2. We will have to keep in mind the other complications that can arise in the healthcare professionals, dentists, who work with the oral cavity, come in contact with the lesions in the oral cavity, can have the infection. The gynecologists or obstetricians who work with the area of the reproductive tract, and if the reproductive tract is having lesions, he or she can have the infections. So this is the broad comparison of both of these diseases based on to the etiological agent, its way of transmission, undergoing the latent stage, the skin lesions and complications. We'll take the herpes simplex virus and its transmission law. And HSV1 is primarily present in the saliva, and infection mostly occur in the face, whereas the HSV2 is infection or transmission through the sexual contact, because the lesions they primarily are found in the genital area. So because the disease is through the sexual contact or with the direct contact with the face or the lesions, HSV1 primarily with the lesions on the face and contact with the saliva, and the HSV2 by sexual contact and contact and lesions on the genital area. Orogenital sexual practices that can also result the vice versa, you see. The asymptomatic shedding of the both HSV1 and 2, it does occur, but HSV2 infections, it markedly increase, whereas the HSV1 is not that much important. The primary infection by 1 in childhood, there is antibody to the HSV2 and does not appear until the age of the sexual activity. So once you see you are mature, you are adult, you will have the antibodies against the HSV1, but not with the 2. The pathogenesis is, you see, the virus after intense, you see uncoat, rigid DNA, and this DNA then integrate with the DNA of the host cells and replicate in the skin or the mucous membrane at the initial site of infections. Now the migration up to the neuron, it occurs with the retrograde axonal flow. And then the organism or the virus, it becomes latent in the sensory ganglionic cells. With HSV1, the latency is in the trigeminal ganglia, whereas in case of 2, it's in the lumbar and sacral regions. Now, during latency, the viral DNA, it is located in the cytoplasm and is not integrated into the nuclear DNA. This is during the latency. But once it reactivates, it then again enters into the nucleus and incorporate with or integrate with the host cell DNA, and then started applying, and it is being released. Now, the reactivation from the latent state by the inducers, the sunlight, the hormonal changes, the trauma, the stress, the fever, that provide the basis for the reactivation of this latent state of viruses. This is what we call as the latent inducers. Then the migration down to the neuron occur, and then the virus replicate in the skin and causes the lesions. So the latent virus, either in the trigeminal ganglion or in the lumbar or sacral regions, it will be reactivated when there is, you see, some inducers like your body is exposed to the sunlight, you have some hormonal changes, you have some trauma, stress, or fever. Then this migration would down to the neurons and would replicate in the skin, and the lesion would be there. Now, the typical lesions, they are the vesicles. And the vesicles, they contain the serous fluid, and this fluid is filled with the virus, virus particles, and the virus and the cell debris. Now, the vesicles, once they rupture, the virus, they're liberated and they can be transmitted to the others. So whenever there is injury to the vesicles, or there is damage to the vesicle, or there is some disruption or rupture of the vesicle, the virus will be released from these lesions and will be transmitted to the others. The multinucleated giant cells, they're typically found at the base of the lesions. If you take the vesicle, you just erode this area, and you make the smear from the base of the lesions, and you stain it with the Giemsa stain or with the methylene blue, what we call as the Zank stains or the Zank smears. You can see the multinucleated giant cells. The type-specific incomplete immunity is there. The reinfection and reactivation, it can occur in the presence of the circulating IgG antibodies. This means that the protection is not complete. The reactivation can occur even in the presence of the IgG antibodies. The cell-mediated immune response is important in the limitation of these herpes viruses. If the immune system is fully functional, the herpes virus infection can be limited. The suppression of the cell-mediated immune response, it results in the reactivation and the spread of severe disease. So if you are immunocompetent, your immunity is fully functional, your cell response is fully functional, you cannot have the reactivation. But once you are immunocompromised, you can have the reactivation and spread of viral infection that may result into severe diseases. Let us see the clinical findings of the herpes simplex one by one. As we said earlier, that the HSV1 causes gingival stomatitis, and you can really see the lesions on the base of the tongue, on the ventral surface of the tongue. Primarily, it occurs in children, and it is characterized by fever, irritability, vesicular lesions in the mouth. And these vesicles, they are full of virus. They are full of virus. So once they rupture, and if a dentist or some of the, you see the ENT specialist is working in this area and bust the lesions, they can get infection of their hand if they're not properly protected. This is what we call the bit loss. Now, involving the entire oral mucosa, tongue, cheeks, lips, you see this is tongue, ventral side, particularly the cheeks, and even to some extent the lips, they are involved. The lesions, they can heal spontaneously in about 3 to 4 weeks time. And this spontaneous healing may provide protection for some time. But if the latency occurs in the trigeminal ganglia, there is every possibility of reactivation of these viruses once you are compromised. Now, the herpes labialis is the characteristic lesion that again appears along with that of the fever. We also call that the fever blisters, or we call that the cold sores that you see most often after the fever has subsided. And you can see the crop of vesicles. Many vesicles at mucocutaneous junctions particularly. You can see over this area, but mostly this area is involved. The mucocutaneous junction is here over also. The mucocutaneous junction is involved on the lips and the nose, and this is how you see the lesions of the herpes labialis there appears. There is tender, itchy papules, and they progress to the vacuoles. They can burst and drain, and there is scab over there that may sometimes to disappear. Recurrence is frequently at the same site has been reported. The sores and fluids, they're highly infectious and should not be touched. So we'll have to be very careful while having these kinds of vesicle on the mucocutaneous junctions of the lips and the nose. Now, the other important is the keratoconjunctivitis. And here you can see the corneal ulcers can occur and the lesions of conjunctival epithelial. Recurrence can lead to the scar formation and brinds. Another important disease that can occur with the help of herpes simplex 1. There is sometimes the scar formation and leads to blindness. Then we have another important clinical finding with HSV1 is the encephalitis that we particularly see in case of what you call the, I'm sorry for this, encephalitis that we can see mostly in case of neonates. It is characterized by the necrotic lesions that is found in the temporal lobe of the brain. There's fever, headache, involvement of the brain may lead to vomiting, seizures, and altered mental status. All those signs associated with the involvement of the brain, vomiting, seizures, altered mental state. This most occur in children. There is acute or protected over several, what you can say, several days, result of either the primary infection or the recurrence. The MRI often reveals the lesions on the brain. The cerebrospinal fluid examination, it typically shows the moderate increase in the lymphocytes, moderate elevations in the proteins and normal glucose levels. Now, keep in mind for the rest of your life that whenever there is viral infection in the brain or in the CSF, you will see the elevation in the proteins, but the glucose normal. The glucose level will remain normal in the viral infection. The CSF glucose readings or numbers remain the same. It will not go high. Whereas in case of bacterial infection, you will find elevated glucose level in the CSF. And the protein, they can also be decreased. The protein increase, glucose level normal, tentatively the viral infection, the protein level decreases, glucose level goes high, probably it is a bacterial infection. The high mortality rate and severe neurological sequences have been reported in the survivor of encephalitis. So if a children has encephalitis because of the HSV1 infections and it stays there, there are chances that the children may have the complications for the rest of its life if he or she survives. Now, other important problem with this HSV1 infection, the hepatic vitro that we just discussed, you can see the skin lesions on the fingers, and this can go up to the hands. And this is mostly, you see, this is observed in case of medical personnel having contact with the patient's lesion, either in the genital area or in the reproductive area or into the hands. And there is a cluster of painful deep vesicles. They can develop in any persons and who carelessly handle his own lesions, active lesions. So you can have it to yourself. If you have lesions and you incidentally or carelessly touch your lesions, which are the active, they have the virus inside, you can get the infection. The herpes gladiatorum, this is what you see in case of wrestlers or another who has the close contact. And this may, it is obvious from the term. The vesicular lesions on the hands, neck, and trunk, they are mostly seen in case of herpes gladiatorum, seen in wrestlers or others who have close body contact with the affected or infected individuals. Now, the eczema herpeticum is another sign that we see with the help of infection with HSV1. This is what we call as the Kaposi varicocilium eruption. You can see the face of these children. The skin infection of this, the atopic dermatitis, vesicular lesions over here, seen at the site of atopic eczema, what we call the eczema. The most cases occur in the children. This is what we call as the eczema. There is skin infection, atopic dermatitis, vesicular lesions, what we call as the eczema. Now, the disseminated infection of herpes 1, there is esophagitis, pneumonia in immunocompromised individuals, patients with T-cell depressed function, they can have this disseminated infection of HSV1. This mostly occurs in immunocompromised individuals or whose cell count is not full or who have undergone the depressed T-cell functions. Now, the diseases are the clinical findings with HSV2. It forms several primary and recurrent diseases. But the most important with HSV2 is the below the belt, and that is the genital herpes. There are painful vesicle lesions that can be seen on the male or female genitalia and even into the anal inner area. The lesions are spheres and they're protected, means that they last for longer than expected time. You suppose that maybe the healing would take place within 1 week time. It may take 5, 6, 7, 8 weeks time. This is what we call as the protected healing. Healings last for longer than the expected time. And the primary disease then in the occurrence. So the primary lesions go for a long time. The recurrence lesions cannot go that long time. So the primary lesions, they are sphere and protected, whereas the secondary reactivated lesions, they are not the protected, and then are their spheres. The primary infection is associated with the fever and inguinal adenopathy in case of genital herpes infection, which is caused by HSV2. The asymptomatic infection in both men prostate, urethra, and in case of women, there's sort of cervix involved. So you can have the asymptomatic infection in case of male in the prostate and urethra, in case of females in the cervix. Then the shedding from the asymptomatic patient, it occurs sporadically and infects the others also. About 80 to 90% herpes genitalis they're caused by the HSV2, and the remaining 10 to 20% are caused by the HSV1 because of the result of the oral genital contact. The suppressive chemotherapy, chemoprophylaxis for the HSV2 lesions be considered, as the lesions caused by HSV2 are more likely to recur than by HSV1. This means that there is more possibility of the reoccurrence of the lesions caused by HSV2, where the lesions caused by HSV1 are not that much recurrence. So we will have to go for the suppressive chemoprophylaxis for the HSV2, because the lesions of HSV2 can reoccur in majority of the cases. Now, the neonatal herpes is another disease caused by the HSV2, and it originates chiefly from the contact with the vesical lesions within the birth canal. So if the mother is infected with the HSV2, the antibodies can also cross, but the mother has the virus in the birth canal, so the baby can acquire this infection from the vesicular lesion present in the birth canal. There is asymptomatic shedding. It can infect the child during birth also. So the newly born baby can get infection either from the lesions already existing or from the virus that is being shed during the process of parturition. Now, the virus varies from severe diseases like the disseminated lesions or encephalitis. If you look at this, we have the lesions in this area. To milder local skin lesions in the eyes and the mouth, and to the asymptomatic infections. The premature infants, which born with the what we call as the cigar burn, you see, it look like that even that as the skin has been burned with the cigar, so it was called the cigarette burn skin lesions. This is very much pattern of the HSV1 infection. So the newly born baby who has been delivered by a mother, that is the frank case of lesions in the reproductive tract, or have the shed of virus in the reproductive tract, get the infection, and you can see sometimes these cigarette burns lesion, which are characteristics of the HSV2 infection. This virus, HSV2 infection, they can be prevented by to this fetus of the contaminated mother, or the carrier mother, or the frank disease mother. We can prevent this kind of infection to the fetus by performing the cesarean section on the woman who have the active lesions, or who have the positive viral culture, or who have the positive antibodies. Now, severe neonatal infections with both 1 and 2, it can be acquired even after the birth from the carriers handling the child. So once you see, even if the fetus or the baby has been born free from the mother, but if it is handled, the fetus is handled, or the baby is handled by the workers who are the carriers, they can get the infection again. There is no congenital abnormalities to any of the significant degrees with the help of HSV2, as we see in case of rubella virus. The rubella or the cytomegalovirus virus, they causes these congenital abnormalities, whereas these abnormalities has not been reported with that of the HSV2 infection. The severe neonatal infections most likely occur from mother who is experiencing the primary herpes infection than the recurrent infection. So if you are the first time receiver of the herpes infections, and you are female and you are going to deliver a baby, there is every possibility that you can have the neonatal infections. But if you are exposed to the recurrent infection, the chances are rare that the fetus would get the infection. There are two reasons for that. Number one, the greater amount of the virus is produced during the primary infection, and that amount is not available in the secondary recurrent infection. And secondly, you see the previously infected mother, it has the IgGs that has caused the placenta and has protected the neonates from the serious disseminated infection. So these are the two reasons why the recurrent lesions or recurrent infections are not that much dangerous for the newly born baby. Here you can see also, you see the aseptic meningitis in children. We have already got from that. There is mild self-limiting fuse squarely, HSV1 and 2. There is erythema multiforme. This is how the erythema multiforme it looks like present on the skin. Appear on the center red area, and then we have a normal skin, and then again outer red area. This is what we called as the erythema multiforme, EM lesions. Center red area surrounded by the normal skin, and then again the skin infected outside. And this is a red ring outside. This is what we call as the target lesions or the bullseye lesions of the HSV2 infection. Now the lesions, they are typically macule, popular. They involve the, you see the tongue, they can involve the hands, and mostly the immune-mediated reaction to the HSV antigen. This is an reaction. It is an autoimmune lesion. It is reaction towards the HSV antigens. Now, the treatment in this case, you see, antiviral drugs like is cyclochlor, useful in preventing the current episode of EM lesions. Drug like sulfonamides and other infectious diseases, including the Mycobacterium pneumoniae, hepatitis B and C, they can also cause these kind of erythema multiformes. Erythema multiforme is, you see, it is also called the Steven-Janssen syndrome. It is characterized by the fever. There is erosive oral lesions, extensive desquamation skin lesions. This is what we call as the erythema multiforme, is also known as the Steven-Janssen syndrome. So you have some fever and the oral erosive lesions and the desquamation of the lesion. Mycoplasma pneumoniae is the most common cause of Steven-Janssen syndrome. So you can have all these lesions in case of Mycobacterium pneumoniae also, just to give you a comparative microbiology clue that which are other bacterial infections that can lead to the formation of erythema. You see the multiformes, or what we call as the Steven-Janssen syndrome, which is really a response of the antigen. Now, the lab diagnosis of the herpes simplex depends upon the isolation of the virus from the lesions, or by growth them into the cell culture, and we can see the cytopathic effect of the CPE within 1 to 3 days. As I told you that the cell culture is not normally adopted in the routine microbiology laboratories. We have to go for the specialized reference laboratory for this culture. Now, identification can also be done with the help of fluorescent antibody staining of the infected skins, or by detecting the virus-specific glycoproteins with the help of ELISA. ELISA, enzyme-linked immunosorbent assay, is another technique that in which we can detect the glycoprotein of the virus, which is specific to the 1 and 2. They can be distinguished by the monoclonal antibodies, which are the antibodies that has been specially designed. They are very, very highly specific antibodies, which have been designed against the HSV1 and HSV separately. And they are, again, you see, designed against the glycoprotein G, and this can also be tested by the ELISA. So if you use the monoclonal antibodies in the ELISA that has been raised against the glycoprotein G, you can have the very, very accurate result for the detection of the disease with the HSV2. Zinc smear, as we have seen, that is made from the lesions from the skin. If there is encephalitis suspected, then the detection of the HSV1 by the DNA in the cerebrous fluid or with the help of PCR. So you can take the spinal fluid out and you can run the PCR and you can detect the genome of the HSV1 in the spinal fluid. The neonatal infection can also be done with the help of viral culture. The PCRS is the most specified and most sensitive, and the highly, you see, the preferred method for the detection of viral diseases. A serological test like the neutralizing test can also be done in the primary infections. It is not used in the reactivated infection because they have the circulating antibodies, so we cannot really get the good result. Excuse me. That was all about the herpes simplex 1, herpes simplex 2, herpes simplex 8, cytomegalovirus, Epstein-Barr virus, and the rest one. I hope you enjoyed your time, and this is very important to keep in mind all the names, and specifically with reference to the attacking area of the HSV1 and HSV2 and the various diseases that are being caused by these infections. Thank you very much.